Hello, everyone. Welcome to our seminar series. For today, we have Gina Nesr presenting her work on singular value decomposition of protein sequences and multiple sequence alignments. Gina is a PhD student in the Department of Biophysics at Stanford University, and she'll be presenting the work done at Johns Hopkins. Gina.
Hi, thanks, Yanyu, for introducing me. Like he said, I'm presenting my undergrad research that I did with one of my best friends and our PI, Doug Barrick, which is singular value decomposition of protein sequences to visualize sequence and residue space. So Autumn and I are co-authors on this paper, but I'll be presenting it today. And our paper is published in Protein Science, published in September. So why SVD for visualizing protein sequences? You can imagine that we have a lot of multiple sequence alignments. You might have them from naturally occurring proteins, or you have them from machine learning based tools. You generate a lot of sequences. So you'll have some amount of sequences and you want to understand what covariance, what relationships do these sequences have with each other. And so one thing you can do is run SVD on a multiple sequence alignment, and you can project them in space. So you end up with a projection like that, and they neatly cluster. We used k-means clustering. And so based on those clusters, you can do some more meta-analysis, and you can understand both the sequences that reside in each of the clusters, as well as the residues that correlate with each of the clusters and signify why they are together by themselves. So just a little bit of lineage background. SVD is something that is typically taught in an introduction to linear algebra course, but many of you may not have taken it. So just some background. Singular value decomposition is the transformation of data into a new coordinate system, and that involves eigenvectors and eigenvalues. An eigenvector is just a set of vectors associated with a linear set of equations. And so an eigenvalue relates those eigenvectors with each other using a constant value. So it's just an integer essentially, or just a numerical value. And so we can relate them with some kind of square matrix, but as I'm going to show you, don't necessarily need a square matrix. So if you have the eigenvector and you multiply it by a square matrix, it's equivalent to the eigenvalue times said eigenvector. So how do we convert a multiple sequence alignment into a coordinate system that is compatible with SVD? Our multiple sequence alignments are sequences with letters representing our amino acids. And so we typically do a one-hot encoding for each of the residues. So in an MSA, you'll have M rows with L columns, which is the length of the protein, and you can convert it. So if you look at the first column, you have lysine, you'll go and look at what its one-hot encoding is, and essentially you'll just replace it. So each residue is now represented with 20 total values, a string of zeros, and 1 representing it. If you have an MSA that comes from naturally occurring proteins, you'll have to do some kind of alignment and clean up the sequences so that they're all the same length. That's pretty typical for an MSA. So you might have a gap residue. And for gap residues, we typically just signify it as a string of just zeros with no one. So it doesn't carry any weight in our SVD. So you have that you have all of those 20 values for each of the residues. So now you have a 20 by L, you have a 20 by M matrix. For the first residue, you'll have a string of 20 integers, 0 or 1, and we consider that the P block. So this block right here is the P block. And P block is a matrix representing the first one-hot encoding residue of each sequence in the multiple sequence alignment. So you can imagine that for every single residue in the sequence, we can concatenate them together, all L of them, and we form what we call the F matrix. So every single residue has its one-hot encoding, concatenate all of that, and you get an F matrix. This F matrix is pretty important. So you have those P blocks and you have this F matrix, but we can also convert it to this other vector representation. You can think about it in a different way, where each row is thought of as a sequence vector. So if you were to look at just the row instead of the block, you'd have the sequence represented as a one-hot encoding binary string. You can also consider with each column, where each column is just a binary residue vector. representing the importance of that residue in that position. And they all have the same shape. It's M by 20L. So what is how do we do singular value decomposition of F? The standard equation for SVD is F is equal to U sigma V transpose. U is just a square matrix. V is a square matrix, but it's transposed. And then our sigma is just the eigenvalues in a matrix. But how do we get those from our one-hot encoded MSA? And that's kind of what our paper explored. And we have the proofs for how you get all of them and how you can think about them related to protein sequences. And I'll go through that from a very brief interface. So what we can first do is form two symmetric matrices, C and D. So we take that F matrix that we just generated from our one-hot encoding, and we can multiply it by the transpose of the F matrix, and that forms C. You can do the reverse, so you can form the D matrix by doing F transpose times that F matrix. So you have now both options. We can think of C as a symmetric sequence identity matrix. And so its shape is M by M. And so in essence, what it is the identity of each sequence within a matrix. And D is the opposite. It's 20L by 20L, still symmetric, but it accounts for a residue pair count. So how often do two values correspond together? So fun fact, if you've ever done principal component analysis or PCA, you actually are using the C and D matrix. In SVD, we're looking at the other two matrices, the U and V matrices from the slide earlier. But if you do PSA, you're actually using C and D. So if you diagonalize them into C and D into symmetric matrices, and then the diagonal matrices, you get this kind of expression here. It's in the paper more rigorously as a mathematical proof, but X and Y are just symmetric matrices, and this caret C is the diagonal matrix. So we know that C is equal to F transpose, and then it's also equal to that X caret C X transpose. And you know that the same is equal for D, and we also have the equation from the SVD equation. So since we know all of that, you can do some substitutions and you can do some rearrangement. And again, that whole proof is in the paper, it's in the supplement. And we walk through it, and so it should be pretty accessible to everyone. You end up realizing that X is just equal to that U matrix, and V is just equal to that Y matrix. And our eigenvalues are equal to our singular values squared. So in other words, our eigenvectors are the columns of U and the rows of V transpose, which come from that SVD equation. And the eigenvalues are just the square of the singular values in sigma. And we can get those very, very simply using either C and D and doing that diagonalization, or using the SVD equation. And so in the paper and then in the code, we explain all of that. In the code, it's done very, very simply. It's just 2 lines of code. And those are also available online to see how you would do that calculation. So now you have that singular value decomposition. And so what ends up happening is that U is a matrix that represents sequence eigenvectors. So each of the eigenvectors is represented by that sequence. And then you have the sigma matrix, and it represents a 0 matrix with minimal, like whichever one's the smallest, M or 20L, singular values along that main diagonal. That main diagonal and those singular values are really important when it comes to representing them and visualizing them in space. And then you have V, and V is a matrix of residue eigenvectors. And so those represent each of the residues within your multiple sequence alignment and what weight they carry. So if we want to extract the kth singular value, and you can imagine that that's the equivalent of looking at which one is the most significant, you have that U is that matrix of sequence eigenvectors. And so for each sequence I, we would just look for that sequence eigenvector, the kth one. And so that's U, like UK. So there's this really nice equation, and it's just that the sequence eigenvector times that residue eigenvector is equal to our singular value times that sequence eigenvector. And so if we relate the sequence and residue vectors that come from the U and V matrices, we know that we have to scale them by the singular value sigma k. And so you end up with these values that you can project into space. So you can plot them on a 2D or 3D coordinate space. And so we can evaluate the sigma k ui k in space, and you have this representation of points, and those represent all of our sequences in space. And so you end up with plots that look like this. Here we have the first singular value plotted against the second singular value plotted against the third singular value, and third singular value plotted against the fourth singular value. If you want to know how you get this equation, you can check also the supplement for the proof. So what about if you want to represent sequences not from an MSA? If you have this new sequence and it's not in your multiple sequence alignment, you can first one-hot encode it, and you get this sequence vector T. It's equivalent to this S vector here. So then you can just plug that into that same equation, where you plug that sequence vector in and you multiply it by your residue eigenvector, or you dot product by the residue eigenvector, and you get this value for each of the singular values, where it's sigma k and then the sequence eigenvector. And so you end up taking that value and just plotting it on top of the space. And so you can project it into the sequence space to examine it relative to other sequences. And so you might realize that, oh, this sequence actually belongs to this set of sequences in the multiple sequence alignment, and you can go do further analyses from there. So one nice thing is that you can also, I don't know why it's frozen. One nice thing is that you can also visualize the residues in your multiple sequence alignment to understand how each of the residues co-vary or relate to one another. So how do we do that? We can rearrange this expression, our SVD equation, into F transpose U, which is equal to V sigma transpose. And so after expanding that, you get a very similar expression, and that's that the residue vector times the sequence eigenvector is just the sigma value, the kth sigma value, times the residue vector. And so in a very similar manner, you can plot all of those values into space. And so you kind of get a residue-based picture of the sequences in each of the singular coordinates. And I'll explain what those colors mean in a little bit. But in this case, we don't really find a need to scale by the singular values. Yeah. So here we did a bunch of toy examples to kind of figure out what each of those singular values meant. And I'll be going through each of these toy models. They're also all in the paper, and we studied them pretty rigorously in there too. So if you consider a sequence, like a protein sequence, that it only has 3 residues, really simple, and each of the possible options are completely different from one another. So for like simplicity's sake, the first position, you're allowed either letter A or letter B. The second position, you're allowed C or D. And the third position, you're allowed E or F. So in the simplest case scenario, there's absolutely no coupling, and everything occurs at equal frequencies. So A occurs at a 50% rate, B occurs 50%, C, D, E, and F. So the probability of each of these sequences is exactly equal, and there's a uniform residue count. And you can see that by this shaded orangey region, and that there is actually that uniform residue pair count. So this is a pretty high entropy model. If you were to take the statistical entropy, it would be a very large value. So you don't actually expect there to be a lot of features or there to be a pattern in the SVD. And that's exactly what we saw. If you look at like the higher ones, there's really no pattern, but we do see some interesting things. So the first singular coordinate here, so VI1, for both the sequence and the residue, is the same for all of the sequences. And so that implies there's some form of conservation happening. And so as we go up, we see some more anti-correlated variation. So here, A and B are heavily anti-correlated, or you can see here that E and F are also anti-correlated equivalently. And then you see that also with the sequences with B and A and D and C. This is a very simple, very naive case, but it illustrates the importance of that first singular coordinate. So first singular coordinate conservation. What about another toy example? What about no coupling and biased frequencies? So we implied some kind of bias between each of the residue positions. And you can see that in this little chart here. So the probabilities of A is 80%, probability of B is 20%, probability of C is 70%, and probability of D is 30%. And you end up getting the probabilities of the sequences here, and then you have the full chart for how likely is everything. So there is some bias in the frequencies, and if you were to go project those using SVD, we see actually considerable variation of the residues in the first singular coordinate. And so you can see that here with the A and B and C and D, and then E and F are also anti-correlated. This correlates with the high frequency of each residue. A is very high frequency in this case, and so is C, and that corresponds with the frequencies that we see in the chart, where the probability of A is 80% and the probability of C is 70%. So you look here, A, and you see that they are actually on the higher end of that singular value. In comparison, B, which is very low and practically white, is on the lower end of that singular value. And we see some more of those like anti-correlated pairs as we move down in the singular coordinates. And then so we see that anti-correlation. C and D are anti-correlated, F and E are anti-correlated, and then equivalently, you can see like the B and the A, or BC and AD are also anti-correlated. What about if we made it a little bit more complicated? What if we added coupling? So we can add pairwise coupling. So we highly prefer B and D to occur together and A and C to occur together. E and F are they're chilling. They're still at that 50% rate. And that is also, you can see what that looks like in the residue pair matrix and in that chart with the probabilities of each of those sequences. And so if we project that all into space, we get charts that look like this. And so there it is again, that conservation along the first singular coordinate. And we see that from our frequencies and our probabilities for each of those sequences and residues. And so there's this mutual dispersion in the higher singular values where B and D always co-occur and A and C always co-occur. And that makes sense because A and C have this higher probability together and B and D have this higher probability. So they will separate, they will disperse, and they'll be they'll separate along those axes in the higher dimensions. And they continue to be like that as you go down the second, third, fourth singular values. And then lastly, what about three-way coupling? So this is a little bit more complicated. The numbers, there's a lot of them here. But you'll have to take my word for it that there is three-way coupling happening here. And so we have two sequences specifically that are very high frequency, and that's ACE and BDF, which are completely opposite. They don't share any of the same residues. And so we see some positive correlation and we see some negative correlation. So A, C, and E are all positively correlated, whereas B, D, and F are all negatively correlated. And in the very high, in the higher, the third and fourth singular values, we see a lot of anti-correlation between everything. And so it forms this really cute circle. We form this really cute circle here. But that is all represented by that three-way coupling. What's really nice is that they all, in the first singular value, they're all dispersed into their own cluster. And so you know that they're all in the same area and the same value. So cool, those are some toy models. So what does that mean in terms of the toy models? Uncorrelated residue biases result in displacement in the first singular coordinate. There's positive covariance between residues at different positions, and this results in mutual displacements in the second singular coordinate. There's also negative covariance happening between residues at different positions, and this results in that opposite displacement. And anti-correlated residues at the same position are also oppositely displaced. So what does that mean with naturally occurring proteins? So we tested it out on 2 different proteins, and I'll introduce them right now. Just their natural multiple sequence alignment. These are also all done in the paper, and everything that we analyze and all of the graphs and all of the code to generate these graphs are all available on GitHub. But we analyzed homeodomain, and it's a relatively small protein. It's 57 residues long, and it's a DNA binding domain, and it's found in eukaryotes. We got the sequences from a website called PFAM, and that's the ID number right there. When we downloaded it at the time, there are about 85,000 sequences, but after we filter for sequence redundancy, so removing highly similar sequences at a 90% cutoff, and then removing any position that had more than 50% gaps, we ended up with about 5,000 semi-unique or somewhat unique sequences. And this pretty homeodomain is this GitHub, or this PDB number. And we also looked at RAS GTPase, and a lot of you are probably familiar with this protein, but it's 160 residues long, and it's involved in signaling pathways for cell growth and apoptosis. And so in a very, very similar manner, we downloaded it from PFAM, the sequences, and there were about 46,000 of them. And after filtering for redundancy, highly similar sequences in those excessive gaps, we end up with about 10,000 sequences. So what does that analysis kind of look like in terms of all of this high amount of sequences? So when we project them in space, we get this really, really nice clustering. And I think it's best represented by these videos. So you can see there's in homeodomain, there's this very unique red cluster, this really nice orange cluster, a blue cluster here, and that green cluster. And they are all very similar in their sequences. And I'll show you what that kind of looks like when you do a more rigorous analysis. But when it comes to RAS, you see a similar thing happening with RAS domain, where it actually forms this kind of pyramidal scheme, and they all cluster on their own into their own separate little niches. And so those are the visualized sequences. What about the visualized residues? So one thing we did is that we did the coloring based on the clusters. And so the color points indicate residues that have frequencies within each cluster enriched by 0.4 or more, or compared to the out of cluster residue frequencies. So those are relatively unique residues that exist in that cluster that don't exist in any of the other clusters. And essentially, these are the residues that distinguish those sequences from the other clusters. And so you actually see that dispersion. There's one residue in the green that completely distinguished everything in the green. There's a subset of red that distinguished everything in that red cluster. And you see that in both homeodomain and in RAS GTPase. So that's pretty fun. That's interesting analysis. What does that actually mean in terms of the protein? So we can actually map those functional features and all of those clusters back to actual proteins and all of those subfamilies that exist for homeodomain and RAS GTPase. These are 2 really pretty good natural test targets because they're so well studied. So for the homeodomain sequences, there's about 84 homeodomains that have been thoroughly characterized and determined, and they've been divided, I think, over the literature into roughly 11 specificity groups. And so we projected those sequences for homeodomain from each of the groups into the different clusters. And so one of the interesting things that came out of it is that 3 of those kind of subfamilies, 6, TGIF, and IR, all clustered into that blue cluster for homeodomain. And those are all types of functional homeodomains that people consider atypical. And so that might mean that these genes represent ancestral families. So that's an interesting analysis. We do see that there is some dispersion between the reds. So there's like some of the BCD actually falls into and trickles into the NK and bar region. But roughly speaking, they're pretty distinct clusters. With RAS GTPAs, which I think a lot of you are familiar with, we see some stronger functional segregation patterns happening. And so if we do a classification scheme, this time relative to like the subcellular processes that each of the GTPAs is involved in, we can classify them into Rho, RAB, and RAS subfamilies and project those projections. So Rho is yellow, RAB is red, and RAS is blue. And then the ARF family sits at that putative vertex around the green. Whereas RAN is at the intersection between ARF and RAB, and those are all represent the ancestry that happened between and the evolution of RAS GTPAs. That's pretty interesting because we did one more analysis, and that's looking at the phylogeny and the taxonomy trees. And you see that as the sequences evolved, the clusters kind of followed along with it, and they were able to segregate both the phylogenetic tree, the sequence evolution, as well as the taxonomic tree. So here's an interesting one, and it's the homeodomain. The red and the yellow all segregate to only metazoa type families, whereas the blue and the green are seen in all of the different families. So that's a pretty interesting thing that we found from here. And these pretty charts were all made from phylo-T, which is a very, very cool way of visualizing both your taxonomy and phylogenetic tree. So thanks to them. So you guys are all here for a machine learning seminar. So what have I been doing with this stuff relative to machine learning? So with ML generated sequences, there are a lot of sequence design methods out there. Most of you are probably familiar with protein MPNN or the sequence designer, and they're pretty non-deterministic. They don't tend to converge very well. So if you generate like 1,000, 10,000 sequences, none of them would ever be the same. I think I did that for one protein, and not a single sequence repeated once. And so often that's why we have to use stuff like yeast library display or design on mini proteins or alpha helical proteins. Alpha helical proteins have a very well-known pattern, while mini proteins are pretty small and they make them a lot easier for us to test and understand, or even just to scan through and understand their structure. And yeast display is really high throughput, so we can test tens of thousands sequences at once and try to see which one worked and which one didn't. So what if we actually kind of understood what machine learning models were doing relative to the sequences? One thing that could do is decrease the amount of sequences we have to try. Maybe it's actually picking up a pattern that we don't want. Or it'll tell us that, hey, it's actually not learning anything. It's very random. We might need to do a better sampling scheme. So I did this I did some like nice little meta-analyses using ML generated sequences that I'm going to present right now. Yeah, so SVD can help us understand what local minimas our models keep finding over and over and over again. Yeah. So I tested on 3 different proteins, and this is just a very fun thing that I've been doing on the side. This is not in the paper. This is just stuff that I've been doing. So this is a pretty cool protein. Tianyu actually suggested it, and has this tri-symmetry. It's like a little bundle. It's 3PGO on PDB, and it's 140 residues long. And so I designed sequences using the JAX version of Protein MPNN by Sergey. It's an incredible version. It's on Google Colab. Highly recommend everyone goes and checks it out. He does a lot of awesome things. And then I just projected those sequences that Protein MPNN spit out into sequence space. So what's pretty neat is that it clusters into 2 very, very distinct clusters. Like there's like a line. You can draw a nice line in between, a nice plane, and it'll separate all of those sequences. Why is it doing that? So to give you kind of an idea, there are 300 blue sequences and about 700 red sequences. So that's like a 1-to-2 ratio. So why? If we project 3PGO onto these sequences that Protein MPNN generated, we actually see that it clusters into the red, but it's not exactly the same. So we can do that residue analysis. We can look at the residues that discern each of them. And so we see that there is indeed one residue. There's like one residue that's red and one residue in the blue sequences. We see one residue in the red and one residue in the blue sequences that actually discern those two clusters. And the rest are all actually pretty the same. And you see that because there is that conservation along the second coordinate. So if we go through and we look at what are those residues, we can pick them out both by the type of residue and the position of them. And so pretty neat is that there's A25, A72, and A19 all occur in that red cluster, and T25, T72, and T19 all occur in that blue cluster. The sequence that comes from 3PGO is the red one. So if you go look at the PDB, A25 exists, A72 exists, and A19 exists in this PDB. So that's pretty neat. You kind of see what kind of pattern and what suggestions it was making from the machine learning generation. So what about something really, really simple? Those alpha helical bundles that a lot of people like to use. So we can generate some sequences from that. And again, I use protein MPNN and project them. And I generated about 4 different clusters that looked about reasonable to me. And there was some enrichment between those clusters. And so we can go through and look at which ones are they. And so in the residues, if we look at which residues they're actually just those capping. They're the capping residues. And that makes sense because I think protein MPNN probably learned that helical bundle because it was in the training set. But there's a lot of flexibility in the capping region for what could possibly work there. So one thing I did was I put this PDB, 7UEK. It's a de novo ovoid tin barrel from Alex Chu and Posu's lab. It just came out. It was dropped into the PDB on the 18th of January. And so this is an oval shaped tin barrel. It's hyperstable. So one thing is that protein MPNN never saw this in the training set. So I thought it would be a fun test set. So I had a design on this ovoid tin barrel and I projected it. And there's absolutely no pattern. It's just a lot of dots and everywhere. And so this is sampled with like the highest trigger. I wanted to minimize the randomness and see if there was actually anything that it was learning significantly. But if you look at the sequence, there's a lot of random choices being made. And so this is 228 residues. It's pretty long. And so most of the multiple sequence alignment looks like this. There's very few, relatively speaking, highly conserved residues occurring in there. So that's interesting in that, hey, maybe actually protein MPNN isn't designing for this protein as well as we'd want. Maybe we need to go in and do more specific searches, or we need to fix some positions, or maybe we need to sample a little bit more rigorously to design sequences that maybe converge at a possible solution, or we can understand what it's trying to do. In most situations, we would probably tackle this by, hey, let's take this, shove it into yeast display, and find which one of these sequences works. But I think in an ideal world, we'd be able to just generate like a handful of sequences, test the handful, and 9 out of 10 of them would work. So some conclusions. SVD provides us a very powerful way to visualize multiple sequence alignment, sequence space, and residue space. And so we can discern both sequence bias, covariance, and coupling based on the singular values that we choose to look at. Analysis of natural MSAs using SVD allows us to cluster, map functional features, and even analyze sequence taxonomy and phylogeny. And this can be pretty useful for identifying new sequences that we find in nature and compare them to the ones that we already have. But we can also apply SVD to ML-generated sequences, and this is only as good as the machine learning models. Models don't often converge, especially for larger or more complicated structures that they haven't seen before. And I'm sure that if you go through, and I've done this before, if you go through and you were to look at very specific parts of your protein and you're analyzing with SVD, you will find some small patterns. but it's an interesting way to kind of pick at the sequences that you're being generated. To reproduce everything except for the ML generated sequence picture images, everything is reproducible from the GitHub. That's the link. It's also linked in the paper. So all of the figures are available. There's Jupyter notebooks. Sorry guys, there's no Google Colab, but there are Jupyter notebooks for you to run everything, and you can go through and modify and do whatever you want with that code. It's all freely available on there. And with that, I'd like to thank my co-author, Autumn. She was incredible in this, and Doug Barrick, who was our advisor in undergrad, who helped us kind of cultivate this into what is now a paper. So that's really exciting. And thank you to everyone else in the Barrick Lab at Hopkins for all their support and all the fun discussions. And then everyone in Wong Lab who has been helping me out with the ML side of things and kind of understanding sequence space and residue space. And then our funding sources for this paper were the NIH and the Institute for Data Intensive Engineering and Sciences. Thank you. And I guess with that, I can take questions.